In this video, I'm going to discuss the HPG, or hypothalamus anterior pituitary gonad axis of both XX, female, and XY, male individuals. As a brief overview before I begin, I want to point out that the hypothalamus releases a hormone called gonadotropin-releasing hormone, GnRH. This triggers the anterior pituitary to release two different gonadotropins, LH, luteinizing hormone, and FSH, follicle-stimulating hormone. LH in the male or XY individual, LH binds to receptors on Leydig cells or the interstitial endocrine cells, triggering them to synthesize testosterone. Testosterone is often converted into DHT by 5-alpha reductase at target tissues throughout the body. FSH binds to receptors on the Sertoli cells, also called the nurse cells or cystinocytes within the testes. The Sertoli cells are triggered by FSH to create androgen-binding protein, which in turn binds to testosterone and stimulates spermatogenesis. Sertoli cells also secrete inhibin in response to FSH and high sperm levels. Inhibin specifically has negative feedback on the FSH secreting gonadotrope cells. Testosterone, and in turn DHT, dihydrotestosterone, exerts negative feedback on both LH and FSH secreting gonadotrope cells as well as the GnRH cells of the hypothalamus. This story is similar in XX individuals, so females, in that GnRH stimulates the anterior pituitary gland to release LH and FSH. LH binds to thecal cells, or the outermost cells of the follicle, which in turn triggers the release of androstenedione, a weakly androgenic derivative of cholesterol. FSH binds to receptors on the granulosa cells, triggering those granulosa cells to convert androstenedione into the highly estrogenic estradiol. Granulosa cells also secrete inhibin, just like the Sertoli cells secreted inhibin. Inhibin has negative feedback on the FSH secreting gonadotrope cells. Estrogen, or estradiol, exerts negative feedback on the anterior pituitary gland as well as the hypothalamus for most of every ovarian or menstrual cycle. The one exception is during the middle of the cycle, so essentially the end of the follicular phase and the beginning of the luteal phase of the ovarian cycle. During that time, estrogen has increased so much as the follicle grows, so as there are more and more thecal and granulosa cells, estrogen surpasses some target threshold and briefly exerts a positive feedback on the hypothalamus and anterior pituitary gland, leading to a massive surge of LH and to a lesser extent of FSH as well. This is going to trigger ovulation, therefore marking the beginning of the luteal phase. After this time, estrogen is going to go back to exerting the negative feedback on the hypothalamus and anterior pituitary gland. The granulosa cells from here on out are going to be called corpus luteal cells, or large luteal cells. They are going to continue secreting estradiol, which has been made from androstenedione of the thecal cells, but also directly synthesizing progesterone. Progesterone with estrogen has a negative feedback on the hypothalamus and anterior pituitary gland. Okay, so that is the overview. At this time, I would like to walk you through the hypothalamus. Here we can see the main hypothalamus, the median eminence. This here is the first capillary bed of the hypophyseal portal system. This is the stalk or infundibulum, and this here is the second capillary bed of the hypophyseal portal system within the adenohypophysis or anterior pituitary gland. We'll start our story up here with the gonadotropin-releasing hormone. Here we can see one single neuron, which actually represents many neurons within several nuclei of the hypothalamus. Usually these axis stories start with these neurons within one particular nucleus release this releasing hormone, but that's not it's not that simple in the HPG axis because a lot of these GnRH releasing neurons are closely associated with olfactory neurons during fetal development. Because the olfactory neurons migrate during development, these GnRH neurons kind of get tugged around with them, and so they're a little bit more widely distributed than other HP axes within the human body. And so a lot of times these GnRH neurons are found in the preoptic area, in the medial basal area, the hypothalamus, partly in the arcuate nucleus, so a little bit more distributed. Okay. As we can see, these neurons release GnRH into the first capillary bed of the portal system. Now, unlike in other HP axes, GnRH isn't just released in response to some kind of a stimulus. It's not released in these huge surges. Instead, GnRH is released at a certain rate and pulses over time every single day. As an adult, these GnRH pulses occur 1 or 2 times every 6 hours. Before puberty, the pulse frequency is a lot more infrequent, and the amplitude, so the actual amount of GnRH, is also much lower than after puberty. And so what might contribute to this change in pulse frequency as well as pulse amplitude is a variety of different stimuli, but some of that is related to what are called kisspeptin neurons. Now, kisspeptin is a peptide hormone, classified as a peptide hormone, that is really important in depolarizing the GnRH cells and therefore increasing the frequency of action potentials. That will ultimately lead to more GnRH in frequency and amplitude. So kisspeptin is really important in triggering this entire process. Now, this can be increased in the time leading up to puberty. And one of the things that has been shown to impact how much kisspeptin is released and therefore how much GnRH is released is actually melatonin. So melatonin, it has been shown, has a negative impact on kisspeptin and therefore on GnRH. And so this is going to decrease leading up to puberty and therefore allowing GnRH to increase during puberty. This is also really important in the seasonality of breeding. So when the days are darker, for example, there will be more melatonin released and therefore less breeding is going to happen. Okay, in non-human species mostly, but there is a little bit of a role in humans as well. Okay, so kisspeptin is really important in generating these pulses of GnRH. Okay. GnRH is going to be transported down to the second capillary bed in the adenohypophysis, and it is going to bind to receptors on gonadotrope cells. If we zoom into these gonadotrope cells, we see that there are actually a couple different types of cells. One type of gonadotrope releases LH, or luteinizing hormone. The other releases mostly FSH, or follicle-stimulating hormone. Now, some gonadotropes release both, but I've separated it out here for demonstration, and also because a lot of the gonadotropes secrete mostly one or the other. As we can see here, a pulse, particularly pulse of GnRH, is ultimately going to trigger the transcription, the translation, and release of luteinizing hormone. Similarly, a pulse of GnRH stimulates transcription, translation, processing, and exocytosis of FSH, ultimately into the blood and out to the rest of the body. Now, each of these molecules is actually made up of 2 subunits. The alpha subunit is the same for both LSH or LH and FSH, and for TSH, thyroid stimulating hormone as well. And so it really is the beta subunit that's going to bind to the receptors and produce the different effects that we saw up here in these 2 summaries. Okay, so in summary, a pulse, particularly of GnRH, leads to the release of LH and of FSH. Now, if GnRH is released in a constant manner, right, so GnRH is just coming in at a relatively constant rate, this actually inhibits the release of LH and FSH gonadotropin hormones. And so if there are fertility issues, you can't just take a pill that is, constantly releasing GnRH. You actually have to make sure that GnRH is released in a pulsatile fashion. Okay? So that is the stimulation of anterior pituitary cells called gonadotropes. There are also a variety of mechanisms that inhibit the release of LH and FSH and therefore will ultimately keep estrogen and testosterone and a variety of other steroid molecules within a target range. The first factor here is GnIH neurons. Okay, so here we can see a couple different neurons ultimately synapsing with different cells in the hypothalamus as well as in the anterior pituitary gland. These neurons are going to release something that's called gonadotropin inhibiting hormone. Now, this hormone was first discovered in birds and later kind of relatives were found in other species as well, including in humans. Now, all of these hormones are a little bit different, but they do function in largely the same manner. And so in humans, the molecule is technically RFRP3. I am just going to refer to this in general as GnIH because they're all having roughly the same role. Now, GnIH has a negative impact on the release of GnRH from the GnRH neurons. And in fact, there is more inhibition of GnRH than there is downstream in the anterior pituitary gland. So here we can see GnIH is going to inhibit the exocytosis of LH, and GnIH limits the exocytosis of FSH as well. Okay. There are also other inhibitory factors. Inhibin. Okay, so just as a reminder, inhibin is released by granulosa cells in XX or female individuals and the Sertoli, nurse, or sustenocyte cells in the male gonads, so the testes. Inhibin specifically inhibits FSH. So as we can see over here, inhibin binds to its receptors on FSH gonadotropes and therefore inhibits the release of FSH. Okay. The estrogen, progesterone, testosterone, and ultimately DHT also have this negative feedback farther upstream. So let's take a look at the gonadotropes in the anterior pituitary first. We can see here that progesterone has a negative impact on the release of FSH and also of LH. So this is going to be really important to keeping these gonadotropins relatively low during the second half of the cycle, that is, in the luteal phase of the ovarian cycle. DHT. DHT is a more potent androgen even than testosterone is. Oftentimes testosterone is converted into DHT by 5-alpha reductase throughout the body as well as within the hypothalamus and anterior pituitary gland. DHT in particular is what is kind of putting the brakes on the release of FSH and LH from the gonadotropes in the male. Okay. And so this is happening in both males and females for the most of the female cycle. Okay. Farther upstream in the hypothalamus, we also see that estrogen and testosterone can bind to their receptors within, not on the surface, but within the GnRH neurons, and therefore make it more difficult to fire action potentials. Right? So it's going to hyperpolarize these neurons and reduce the release of GnRH1. Okay. Estrogen and testosterone can also inhibit the pulse generator. Right? So it's going to, again, not depolarize the neurons and therefore reduce the action potential frequency. Okay. So again, this is how these steroid hormones are going to exert negative feedback farther upstream. Now, during the mid-cycle surge of the female, so right before ovulation, this negative feedback essentially reverses and is acting as positive feedback. And so there's some threshold level of estrogen that reverses this process. And while this isn't completely understood to my knowledge, there are a few things that we do know about this reversal of the feedback. Okay. First of all, there is what is called a surge center, which is likely in the rostral paraventricular region of the third ventricle. Okay, so right in the hypothalamus there, next to the third ventricle. And these particular kisspeptin neurons within the surge center are responsive to a particularly high level of estrogen. What this is going to do is it's actually going to exert a positive stimulus on the kisspeptin neuron, therefore releasing more kisspeptin, therefore releasing bigger and more frequent surges of GnRH, and therefore releasing LH and FSH in a much higher abundance. Now, LH in particular is released in response to more frequent pulses of GnRH. Right? So the faster pulses are going to release more LH, slower pulses are going to release more FSH. Right? So there's actually an ability to, differentially secrete one versus the other. Faster is more LH, slower is more FSH. Okay, so during this mid-cycle surge, which ultimately triggers ovulation, high levels of estrogen is going to trigger more kisspeptin, bigger, stronger pulses of GnRH, and so there's going to be a lot more LH that is secreted, which actually stimulates ovulation, and to a lesser extent, FSH. Okay. Another piece to this puzzle is that in addition to the release of GnRH, another hormone called galanin is released as well. And so galanin, in association with a particularly high level of estrogen, again, some threshold value, together, these two hormones are going to enhance the response of these LH secreting gonadotropes to the larger GnRH pulses. And so this is just one more way where you get this surge of LH on top, or kind of in opposition to the normal negative feedback on LH during the rest of the cycle. Okay, so in summary, GnRH is released in pulses in response to, partly anyway, the release of kisspeptin from neurons within the arcuate nucleus. GnRH pulses trigger the release of LH, particularly when the pulses are big and fast, and also LH, which is stimulated more by slightly slower and smaller pulses of GnRH. Both of these hormones, LH and FSH, are going to be dumped into the blood and ultimately are going to trigger the synthesis of steroid hormones farther downstream, as well as spermatogenesis and the development of the follicle in the female. Negative feedback is exerted on the anterior pituitary cells, the gonadotropes, by GnIH, as well as inhibin and different steroid hormones. Negative feedback on the hypothalamus is exerted by GnIH neurons, a lack of kisspeptin, an abundance of steroid hormones directly on GnRH, and all of this is happening for the most part in males and females, except for mid-cycle, right, mid-ovarian / menstrual or uterine cycle, when high levels of estrogen promote this surge in kisspeptin, GnRH, lots of LH, and to a lesser extent, FSH. So once again, this has been a summary of the HPG, hypothalamus anterior pituitary gonadal axis of both XX and XY individuals. Thank you so much.